Hello everyone. Welcome to today's presentation. I'm excited to explore with you the fascinating topic of how do organisms reproduce. On this first slide, we're just getting started. We'll be looking at the different ways living things create new life, the processes involved, and why reproduction is so essential for all living organisms. So, let's dive in and discover the amazing diversity of reproductive methods in the natural world. Let's take a moment to look at the overall structure of our presentation. We start with the basic concept of reproduction, which is essential for the survival of all living organisms. Moving forward, we explore the various types of reproduction, including sexual and asexual methods. We then delve into the different processes involved in reproduction, such as DNA copying, fertilization, and the formation of seeds. These are crucial steps that ensure genetic continuity. Next, we focus on human reproductive systems, highlighting the functions of male and female reproductive organs and the importance of reproductive health. We also examine plant reproductive systems, including the female and male parts, pollination, and fertilization in plants. A significant part of our discussion covers vegetative propagation, a type of asexual reproduction that allows plants to reproduce through parts like tubers, stems, or leaves. This includes natural methods like fragmentation and regeneration, as well as artificial techniques like grafting and budding. We also look at the importance of hormones in regulating reproductive processes and population control measures to maintain ecological balance. Throughout the presentation, you'll notice activities like observing spirogyra, bread mold, and yeast, which help us understand these concepts in a hands-on way. Finally, we emphasize the significance of reproductive health and how understanding these processes helps us appreciate the diversity and adaptability of living organisms. This overview connects all these topics together, showing how reproduction is fundamental to life and its continuity. Now, let's look at the transfer of variation from one generation to the next. This process is essential because it helps maintain the size of the population over time. Without this transfer, populations could diminish or fail to adapt to changing environments. This continuation of life on Earth depends heavily on the ability of living organisms to produce new individuals of their own kind. This ongoing production ensures that life persists generation after generation. And this whole process of producing new individuals is what we call reproduction. It's a fundamental concept because it links directly to the survival and continuity of species. So, to sum up, reproduction is key not just for individual organisms, but also for the stability and evolution of life on our planet. DNA in the cell nucleus serves as the information center for making proteins. It contains all the instructions our cells need to function properly. The first step in reproduction is copying this DNA, a process called replication, which prepares the cell for division. After DNA replication, the cell divides to produce new cells. It's important to remember that variations can occur during the copying process. These small differences, or variations, are actually the foundation of evolution. They introduce diversity within a species, which is crucial for adaptation and survival over time. In fact, these variations can be beneficial, helping species to survive changing environments and challenges. So, understanding DNA replication and the resulting variations gives us insight into how life evolves and continues to adapt. This connection between genetic copying, variation, and evolution is a key theme in understanding biological reproduction. Any change in the DNA causes a change in how proteins are synthesized, and this, in turn, affects the body's design. Essentially, this means that even small variations in DNA can lead to differences in characteristics. Think of DNA as the blueprint for constructing the body. It's what ensures that our body develops correctly and maintains its characteristics across different generations. This consistency is crucial because it allows traits to be passed down reliably from parents to offspring. That's why DNA copying is so important in reproduction. It ensures that each new organism receives an accurate copy of the genetic information. Without this precise copying process, the characteristics we inherit could become inconsistent, which could affect the survival and functioning of the species. So, understanding DNA copying helps us appreciate how traits are preserved and transmitted across generations, maintaining the continuity of life. Variation refers to changes in the body design of an individual. These changes are crucial because they allow organisms to adapt and survive in a variety of habitats or niches. Small variations, or gradual changes over time, can really boost a species' chances of thriving under different conditions. It's like giving them more tools to cope with their environment. However, it's important to remember that drastic variations in a single individual can actually reduce their chances of survival. Too much change too quickly can be risky. This concept of variation is fundamental because it forms the basis of evolution, the process through which species gradually change over generations. Ultimately, variation is beneficial for the species as a whole, helping it adapt and persist in a constantly changing world. Now, let's explore the two main types of reproduction. First, we have asexual reproduction. In this type, a new individual is produced from just a single parent. This means there's no need for a partner or the union of gametes. Because it involves only one parent, the offspring are usually genetically identical to the parent, which helps maintain specific traits. However, one thing to note is that there is very little genetic variation in this process, so the offspring tend to be clones of the parent. On the other hand, we have sexual reproduction. This process involves two parents, and the new individual is formed when their gametes, sperm and egg, union. Because of this combination, the offspring are not exactly the same as either parent, which introduces genetic variation. This variation is crucial for evolution and adaptation over time, making populations more resilient to changes in their environment. So, in summary, asexual reproduction is quick and results in identical offspring, while sexual reproduction promotes diversity but requires more time and energy. Both types play important roles in different organisms and contexts, fitting into the bigger picture of how living things reproduce and pass on their traits. Now, let's take a closer look at the various types of asexual reproduction. First, we have spore formation, which is a common method among fungi and some plants. Spores are typically produced in specialized structures and can survive harsh conditions, allowing the organism to spread and grow in new locations. Next, there's vegetative propagation. This is a form of asexual reproduction seen in plants, where new individuals grow from parts of the parent plant, like stems, roots, or leaves. It's a quick way for plants to reproduce and spread without the need for seeds. Regeneration is another fascinating process, especially in animals like starfish or certain worms. If a part of the organism is lost or damaged, it can regenerate into a complete new individual, essentially regrowing the missing parts. Fragmentation involves the organism breaking into pieces, each of which can develop into a new, complete organism. This method is common in some invertebrates and simple plants. Budding is a process often seen in yeast and hydra, where a new organism grows out of the body of the parent. Once it's developed enough, it detaches and lives independently. Finally, fission, which is a straightforward division of the parent cell into two or more daughter cells. This is especially common in bacteria and some protozoa, where the cell simply splits into identical copies. All these methods highlight how organisms can reproduce efficiently and rapidly without the need for sexual processes, supporting survival and propagation in various environments. Let's talk about fission, which is a common method of reproduction in single-celled organisms. For example, Plasmodium, the parasite that causes malaria, reproduces through fission. Now, this process typically occurs during unfavorable conditions, when the organism needs to quickly increase its numbers to survive tough environments. In such cases, the cell undergoes fission into many smaller cells, a process known as multiple fission. Think of organisms like amoeba and paramecium, which use this method. Their cells divide into numerous daughter cells during these challenging times. On the other hand, when conditions are favorable, meaning the environment is suitable for growth. The organisms usually reproduce through binary fission. This is a simpler process where the cell divides into two identical daughter cells. So, in summary, fission is an essential reproductive strategy that allows single-celled organisms to adapt and survive in varying conditions by either producing many cells during tough times or just two during good times. It's a remarkable example of how life adapts to environmental changes. Let's take a look at budding, which is another fascinating method of reproduction in some organisms like Hydra and yeast. Essentially, budding involves a small protrusion, or a bulge, developing on the body of the mature organism. This bulge grows and matures until it's fully developed, almost like a mini clone of the parent. Once it reaches maturity, it detaches from the main body and becomes independent. This process allows these organisms to reproduce quickly and efficiently, often without the need for sexual reproduction. It's a great example of asexual reproduction, and it highlights how some creatures can rapidly increase their population through this method. Understanding budding helps us see the diversity of reproductive strategies in the biological world, which is very much in line with the overall theme of the presentation, exploring different ways life persists and propagates. Now, let's talk about fragmentation. This is a fascinating form of asexual reproduction where an organism's body simply breaks into smaller pieces. Each of those pieces has the potential to grow into a new, complete individual. Think of it as if the organism is splitting itself up, and each part can regenerate into a new organism. It's important to note that this process happens upon maturation, meaning once the organism is fully developed, it can produce these fragments. This method is seen in various organisms, like certain types of worms, starfish, and even some plants. It highlights how diverse reproductive strategies can be, especially in the context of asexual reproduction. Fragmentation allows these organisms to quickly increase their numbers without the need for sexual reproduction, which can be advantageous in stable environments. So, overall, fragmentation is a simple yet effective way for some species to reproduce and survive. Regeneration is the incredible ability of an organism to replace or repair any lost part. It's a fascinating process that shows how resilient living things can be. This process is carried out by specialized cells that are capable of transforming and multiplying to rebuild damaged or missing tissues. For example, organisms like Planaria and Hydra are well known for their remarkable regenerative abilities. Understanding regeneration not only highlights the adaptability of these creatures, but also connects to the broader theme of how organisms maintain their integrity and survive injuries. This ability is a key aspect of biological resilience and evolution. Let's take a closer look at the differences between fragmentation and regeneration. These two processes are fundamental in understanding how organisms grow and repair themselves. Fragmentation involves breaking into parts, where each part has the potential to develop into a new organism, essentially a form of asexual reproduction. Think of it as cutting a starfish, and each arm can grow back into a whole new starfish. On the other hand, regeneration generally refers to the process of restoring lost or damaged tissues or organs. It's about repair and regrowth within an existing organism, like how lizards can regrow their tails. The key difference here is that fragmentation results in new, independent individuals, while regeneration is about restoring the original organism. Recognizing these differences helps us understand the diverse strategies life uses to survive and thrive, which ties back to our overall theme of biological adaptability and resilience. Now let's look at some examples like Aspergillus and Rhizopus, which are fungi or even mushrooms. These are all part of the plant kingdom, specifically within the fungi group. An interesting point here is that sporangia, which are special structures, play a crucial role in how these organisms disperse their spores. These spores are unique cells that develop inside the sporangia, and from there, they can grow into new plants or fungi, completing their life cycle. What's really fascinating is that in some cases, these spores can give rise to an entirely new organism, meaning the sporangia essentially have the power to create a whole new plant or fungus from just a single spore. This process of spore formation is a key way these organisms reproduce and spread, ensuring their survival and proliferation in various environments. So, understanding spore formation helps us see how these fascinating organisms continue to thrive and expand across different habitats. Let's look at some examples like bryophyllum, rose, jasmine, and sugarcane. These plants are interesting because they can reproduce through their vegetative parts. When I say vegetative part, I mean the non-reproductive or non-flowering parts of the plant, such as the stem, root, or leaves. Vegetative propagation is a fascinating method of reproduction because instead of producing seeds, a new plant can grow directly from these vegetative parts. This process is often quicker and more reliable than seed formation, especially in horticulture and agriculture. So, the key idea here is that vegetative propagation involves a vegetative plant part, like a stem, root, or leaf, giving rise to a new, independent plant. This method is widely used because it allows plants to reproduce efficiently and maintain their desirable traits. It's an important concept that ties into how plants can be propagated outside of natural seed production, which is especially useful for cultivating certain plants. Artificial vegetative propagation is a fascinating method used by humans to reproduce plants efficiently. It involves techniques like cutting, layering, and grafting, which allow us to create new plants that are genetically identical to the parent. For example, when we talk about cuttings, this method is commonly used with plants like grapes, bougainvillea, cactus, and pineapple. These plants can be propagated quickly and easily by taking a part of the plant, such as a stem or leaf, and planting it to grow a new, healthy plant. This approach is especially useful for plants that do not produce seeds readily or when we want to preserve specific desirable traits. Overall, artificial vegetative propagation plays a crucial role in agriculture and horticulture, enabling us to multiply plants efficiently and maintain the quality of the crops we rely on. Now, let's move on to layering, specifically mound layering. This technique is quite interesting because it allows the new plant to grow independently from the main plant. Essentially, you make a mound of soil around a part of the stem, encouraging roots to form at that point. Once the roots are established, you can cut the new plant away from the parent, and it will continue to grow on its own. It's a practical method for propagating certain plants efficiently. This technique is a part of artificial vegetative propagation, which is all about human intervention to multiply plants. It's especially useful when we want to produce new plants that are genetically identical to the parent. Overall, mound layering is a simple yet effective way to propagate plants, connecting directly to our theme of how humans can propagate plants through various techniques to meet horticultural and agricultural needs. Let's take a closer look at grafting, which is a fascinating technique in artificial vegetative propagation. When we graft, we're essentially joining two different plant parts to create a new, functional plant. The key idea here is that the scion is taken from a desirable plant, one that has superior qualities like better fruit, flowers, or growth habits. The root stock, on the other hand, provides the support and a strong root system, often chosen for traits like disease resistance or adaptability to soil conditions. An important detail is that the cambium layers of both the scion and the root stock must be in direct contact. This cambium layer is the growing part of the plant just beneath the bark, and for successful grafting, these layers need to align perfectly to enable the tissues to grow together and form a union. Once the grafting process is successful, you end up with a new plant that combines the root system of the root stock with the shoot system of the scion. This means you get the best of both worlds, robust roots and desirable above-ground qualities. Grafting is a form of artificial vegetative propagation, a technique that allows us to reproduce plants with specific traits more quickly and reliably than through seed propagation. It's widely used in horticulture and agriculture to improve crop quality and productivity. Sexual reproduction involves both male and female individuals coming together to produce new organisms. It's a process that requires the formation and eventual fusion of specialized cells called gametes, which are the male and female reproductive cells. This fusion creates a new organism with a unique combination of genes. One of the most important outcomes of sexual reproduction is variation among individuals. This variation is crucial because it helps species adapt to changing environments over time. Now, an essential part of this process is cell division, specifically meiosis. Meiosis reduces the chromosome number by half and ensures that when the gametes fuse, the resulting organism has the correct number of chromosomes. This whole process highlights how sexual reproduction not only creates new life, but also promotes diversity, which is key to survival and evolution. Variations ensure the retention of a species within the community. When we talk about variation, we're referring to the differences that arise within a species, which are crucial for its survival. Since DNA from two individual organisms of the same species is involved, sexual reproduction naturally results in variation. This process of crossing over during sexual reproduction is essential because it shuffles genetic material, creating new combinations. These variations are vital because they enable organisms to adapt to different environments. Think of it as a way for species to evolve and thrive in diverse habitats. During this process, the fusion of gametes, sperm and egg takes place, which is fundamental in maintaining a constant number of chromosomes in each generation. This balance ensures the stability of the species' genetic makeup over time. Overall, sexual reproduction plays a key role in generating diversity, which is the foundation for adaptation and survival in a constantly changing world. Today, we're looking at the plant reproductive system. The female reproductive part of a flower is called the carpel, which plays a crucial role in producing seeds. The male reproductive part is known as the stamens, responsible for releasing pollen. Remember, the flower itself is the reproductive structure of the plant, bringing these parts together for reproduction. Understanding these components helps us see how plants create new life and continue their species. Let's take a closer look at the parts of the stamen, which is the male reproductive part of a plant. The stamen plays a crucial role in plant reproduction by producing pollen, which contains the male gametes. It typically consists of two main parts, the anther and the filament. The anther is where the pollen is produced and stored, and it's often located at the top of the filament. The filament, on the other hand, is a stalk that supports the anther and positions it in a way that facilitates pollination. Understanding these parts helps us appreciate how plants reproduce and ensure the survival of their species. So, the stamen is essentially the plant's male reproductive system, working behind the scenes to enable fertilization and the production of new plants. Now, let's take a closer look at the parts of the carpel, which is the female reproductive part in plants. The carpel plays a crucial role in plant reproduction, and understanding its structure helps us appreciate how plants reproduce sexually. The carpel typically consists of three main parts: the stigma, the style, and the ovary. The stigma is the sticky top part that captures pollen. The style is the tube-like structure that connects the stigma to the ovary, providing a pathway for pollen to travel. And finally, the ovary, which is located at the base, contains the ovules, or the eggs. When pollen reaches the stigma, it germinates and grows down through the style to reach the ovules in the ovary, leading to fertilization. Recognizing these parts and their functions helps us understand the complex process of plant reproduction and how flowers ensure the production of seeds for next generations. Today, we're going to talk about pollination, which is a crucial part of plant reproduction. There are two main types of pollination: cross-pollination and self-pollination. Cross-pollination happens when pollen grains are transferred from the anther of one flower to the stigma of another flower, often with the help of agents like wind, water, or insects. This process promotes genetic diversity, which is beneficial for the health of plant populations. On the other hand, self-pollination occurs when pollen from a flower fertilizes the same flower or another flower on the same plant. This can happen naturally without external agents. Both types of pollination are essential for different plants, and understanding these processes helps us appreciate how plants reproduce and survive. Now, let's look at the different types of pollination. First, we have self-pollination, which occurs when pollen grains are transferred from the anther to the stigma of the same flower. This means the flower essentially fertilizes itself, which can be advantageous for plants in stable environments. Next, we have cross-pollination, where pollen grains are transferred from the anther of one flower to the stigma of a different flower. This can happen between flowers of the same plant or even different plants of the same species, promoting genetic diversity. Looking at the table, you can see that when pollen moves from flower A to the same flower A, that's self-pollination. When it moves from A to B or A to C or B to C, that's cross-pollination. Understanding these types helps us appreciate how plants reproduce and ensure their survival through different mechanisms. Fertilization in plants is a fascinating process, and it all starts with the fusion of two vital cells. On one side, we have the male gametes, which are the male germ cells, and on the other, the female gametes or egg cells. When these two come together, fertilization occurs, leading to the formation of a new plant embryo. This process is crucial because it ensures the continuation of the plant species and contributes to genetic diversity. Throughout this presentation, we've seen how fertilization is a key step in plant reproduction, connecting the development of flowers to the creation of seeds and new life. Remember, the fusion of these gametes is what kickstarts the cycle of growth and reproduction in plants. Now, let's take a closer look at the formation of a seed. This process is truly fascinating because it represents the beginning of a new plant's life cycle. Essentially, it all starts with the fertilization of the ovule, where pollen meets the ovule inside the flower. Once fertilization occurs, the ovule transforms into a seed. This seed contains all the necessary genetic information and stored nutrients to support the initial growth of the plant. The formation of the seed is a complex yet efficient process that ensures the survival and propagation of plant species. Understanding this process highlights how plants have evolved mechanisms to reproduce and thrive in diverse environments, which ties back to our overall theme of plant adaptation and survival. Now, let's move on to slide 28, which focuses on the human reproductive system. As we've discussed earlier, puberty marks the period when the reproductive systems of young organisms become functional at a specific age. In humans, this is when the body starts to develop the ability to reproduce. The reproductive system includes organs like the testes in males and ovaries in females, which produce sperm and eggs, respectively. These organs also produce hormones that trigger secondary sexual characteristics, such as facial hair in males or breast development in females. Recognizing how these systems develop is key to understanding human growth and development, and it ties into the overall theme of how organisms mature and become capable of reproduction at a certain stage in life. Now, let's talk about the importance of hormones. First, hormones play a crucial role in the development of mammary glands, which are essential for breastfeeding and nurturing infants. They also cause changes in the pitch of the voice, particularly during puberty, giving it that deeper or more resonant quality. Hormones are responsible for the growth of facial, axial, and pubic hair, which are secondary sexual characteristics that distinguish males and females. These features develop because of the influence of specific hormones, mainly androgens and estrogens. Furthermore, hormones help develop and maintain the structure and function of accessory sex organs, such as the testes, ovaries, and related structures, ensuring they work properly. They also regulate gametogenesis, which is the process of forming gametes, sperm in males and eggs in females, essential for reproduction. Overall, hormones are vital because they orchestrate these physical changes and reproductive functions, linking the development of secondary sexual characteristics to the overall process of human reproduction. They are truly central to our sexual development and reproductive health. Let's take a closer look at the male reproductive system. First, you can see the pair of testes, which are housed inside a pouch-like muscular structure called the scrotum. This setup helps regulate temperature, which is vital for sperm production. Next, we have the vas deferens, the tube that transports sperm from the testis to the urethra. Moving on, the penis is the external genital part, with an opening that allows for the passage of semen during ejaculation. Inside, there are several important glands, starting with the seminal vesicles, which produce a fluid that nourishes and helps sperm move. The urethra runs through the penis and serves as a passage for both urine and semen, but not at the same time. The prostate gland surrounds the urethra and adds additional fluid to semen, aiding in sperm motility. The urinary bladder stores urine and is part of the urinary system, but closely related to the reproductive structures. The ureters are tubes that carry urine from the kidneys to the bladder. When we look at these components together, we see how the reproductive and urinary systems are interconnected, working in harmony to perform their vital functions. Sperms are produced in the testis, which is the primary male reproductive organ. From there, they enter the urethra through a long tube-like structure called the vas deferens. The vas deferens transports sperms from the testis to the urethra. The urethra then opens outside of the body through the genital pore, allowing the sperms to exit during ejaculation. The urethra is enclosed within the penis, which is a muscular organ that helps in delivering sperms outside. Just before sperms enter the urethra, they are enriched with secretions from the prostate gland and seminal vesicle. These secretions provide nutrients and help in the motility of sperms, making them more capable of fertilizing an egg. It's important to note that sperms require a temperature about 1 to 3 degrees Celsius lower than the normal body temperature for optimal production and survival. To maintain this cooler environment, the testes are located in the scrotum, which is a muscular pouch-like structure outside the main body cavity. The urethra serves a dual purpose. It's a common passageway for both urine and sperms, depending on the situation. The placement of the testes in the scrotum not only helps regulate temperature, but also provides an environment conducive to sperm development. In summary, the testes produce sperms, which are then transported through the vas deferens to the urethra, enriched with secretions from accessory glands, and finally expelled outside through the genital pore, all while maintaining the necessary temperature for healthy sperm development. Now let's take a closer look at some of the key structures of the female reproductive system. First, we have the uterus, which is a vital organ where a fertilized egg can develop into a fetus. Next, there's the vagina, which serves as the canal leading from the external body to the uterus. It plays a crucial role in sexual intercourse, childbirth, and menstrual flow. Moving on, we see the funnel-like structure leading to the fallopian tubes. These tubes are essential because they are the site where fertilization typically occurs, as the egg travels from the ovaries to the uterus. Speaking of which, the cervix is the lower part of the uterus that connects to the vagina, acting as a gateway between the two. Lastly, we have a pair of ovaries, which are responsible for producing eggs and releasing hormones like estrogen and progesterone. These components work together harmoniously to support reproduction and hormonal balance. Understanding these structures helps us appreciate the complexity and beauty of the female reproductive system as a whole. Let's take a closer look at the functions of the female reproductive system. The pair of ovaries are essential because they produce ova, which are the eggs necessary for reproduction. The ovaries are connected to the uterus, which is the central reproductive organ. The uterus is where a fertilized egg can develop into a baby. The cervix and vagina are also important parts. The cervix acts as a gateway between the uterus and the vagina. The uterus opens into the vagina through the cervix, allowing for menstruation, sexual intercourse, and childbirth. The uterus also secretes two vital hormones, estrogen and progesterone, that regulate the reproductive cycle and support pregnancy. At the end of each ovary, there's a funnel-like structure called the fallopian tube. This tube captures the ovum released during ovulation and provides the pathway for it to reach the uterus. The vagina itself has a tubular structure, which serves multiple functions, including acting as the birth canal through which the baby is delivered. Inside the ovaries, there are follicles that produce ova, especially starting from puberty. This is when the ovaries begin releasing eggs regularly. The fallopian tubes from both ovaries open into an elastic, bag-like structure called the uterus, where fertilization typically occurs and the embryo develops. It's also important to note that the vagina is separate from the urethra, which is the tube through which urine is expelled. This separation is crucial for the proper functioning of both systems. Overall, the female reproductive system plays a vital role in reproduction, hormone production, and supporting pregnancy, all connected in a beautifully coordinated way. If fertilization does not occur, the process takes a different turn. The uterine lining, along with the blood vessels supporting it, breaks down. This broken-down lining then moves out of the body through the vagina, which is what we recognize as bleeding during a period. Meanwhile, on the ovarian side, follicles in the ovary mature into an egg or ovum. Once they mature, these eggs are released from their respective ovaries. At the same time, the uterine wall begins to thicken in preparation for a potential pregnancy. The ovum waits in the fallopian tube, hopping to meet sperm and be fertilized. If fertilization doesn't happen, the uterine wall continues to thicken initially, but ultimately it will shed during the menstrual flow. This cycle, which we're discussing here, is a fundamental part of the female reproductive system and highlights the intricate coordination between the ovaries and uterus. Now, let's talk about the placenta, which is a special tissue that forms between the uterine wall and the developing fetus. The placenta plays a crucial role in supporting the growth of the baby. It provides essential nutrients to the embryo, ensuring it gets everything it needs to grow properly. Additionally, the placenta acts as a kind of exchange station, helping in the transfer of carbon dioxide and oxygen between the mother and the fetus. It also helps remove waste substances produced by the developing baby, keeping the environment clean and healthy. Another vital function of the placenta is that it helps in attaching the fetus securely to the uterine wall, providing stability throughout the pregnancy. It's important to remember that the development of the fetus takes about 9 months, and during this period, the placenta is constantly working to support and protect the growing baby. Finally, when it's time for birth, the process occurs through rhythmic contractions of the muscles in the uterus, which help to push the baby out into the world. The placenta, therefore, is essential not only for nourishment and waste removal, but also for the entire process of pregnancy and birth. AIDS, or acquired immunodeficiency syndrome, is caused by the human immunodeficiency virus, commonly known as HIV. As of now, there is no cure for AIDS, and if it's not treated properly, it can lead to death. It's a serious condition that highlights the importance of prevention and early detection. Most sexually transmitted diseases, or STDs, tend to cause specific symptoms like a burning sensation during urination and urethral discharge. These symptoms are signs that something's not right and need medical attention. Some common examples include gonorrhea, syphilis, trichomoniasis, and of course, AIDS itself. STDs are infectious diseases caused by various pathogens, bacteria, protozoans, viruses, and fungi, that spread through sexual contact. This makes awareness and prevention crucial in controlling their spread. Focusing on sexually transmitted diseases, it's essential to understand how we can prevent and control them effectively. Education plays a key role here, knowing how these diseases spread and how to protect ourselves. Along with prevention, awareness about fertility regulation methods is vital. It's about understanding your rights, maintaining the freedom to make choices about reproduction, and exercising that right responsibly. Having a responsible, safe, and satisfying reproductive life is a goal for everyone. It involves making informed decisions, practicing safe sex, and being aware of reproductive health issues. Reproductive health encompasses many aspects. It's not just about avoiding diseases, but also about ensuring a healthy, balanced approach to sexuality and reproduction. It's about empowering individuals to make choices that enhance their overall well-being and quality of life. Today, we're going to demonstrate asexual reproduction through budding and yeast. First, you'll need some materials: sugar, water, a test tube, yeast granules, cotton, a slide, and a cover slip. Start by adding a pinch of yeast granules into the test tube. Then, place a cotton plug on top and keep the test tube in a warm place to allow the yeast to activate and reproduce. Next, prepare a sugar solution by dissolving 10 grams of sugar in 100 milliliters of water. From this solution, take about 20 milliliters and transfer it into a test tube. Now, take a small drop of this yeast suspension from the test tube and place it on a slide. Observe this under the microscope to see the budding process in action. This activity is a great way to visualize how yeast reproduces asexually through budding, which is a common form of reproduction in many single-celled organisms. It highlights how new cells form as outgrowths or buds from the parent cell, which eventually separate. By understanding budding in yeast, we get a clearer picture of asexual reproduction's efficiency and simplicity, connecting to the broader theme of how organisms reproduce and sustain themselves. So on this slide, we're observing that yeast primarily reproduces asexually through a process called budding. What that means is that a new yeast cell forms as a small outgrowth on the parent cell, then gradually enlarges until it separates. This method allows yeast to multiply rapidly and efficiently, especially in favorable conditions. It's a great example of a simple yet effective reproductive strategy in microorganisms. Understanding this process helps us see how yeast can quickly colonize environments, which is important in both natural ecosystems and industrial applications like baking and brewing. So, in the bigger picture, yeast's asexual reproduction highlights how organisms adapt their reproductive methods to optimize survival and growth. Now, let's take a closer look at bread mold growth. As you can see, it starts by growing directly on the bread. Initially, the mold appears white, which is the early stage of its development. As it matures, you'll notice the color changing to brown and eventually turning black. This color change indicates different stages of growth and maturity. Observing these changes helps us understand how mold develops over time. This activity will give you a hands-on experience of seeing these growth stages yourself. Remember, mold is a type of fungus that thrives in moist, warm environments, which is why bread left out can become a perfect spot for it to grow. So, paying attention to these color changes can also help us understand the life cycle of mold and its impact on food. Let's take a closer look at this slide. Here, we see an image of an amoeba, which is a simple, single-celled organism. The first picture shows a typical amoeba, just one cell doing its thing. Now, the second image highlights a larger amoeba undergoing a process called binary fission, and you can see a constriction in the middle. This constriction is really important because it marks the beginning of the cells splitting into two. Binary fission is essentially how these organisms reproduce asexually, by dividing into two identical daughter cells. It's a straightforward but efficient way for amoebas to multiply, especially in favorable conditions. As we move through this presentation, understanding binary fission helps us grasp how simple organisms like amoebas maintain their populations and adapt to their environments. So remember, the key point here is that binary fission is a form of asexual reproduction, characterized by this visible constriction, leading to the formation of two new amoebas from one. Now, let's take a closer look at the Spirogyra filament under the microscope. As you observe, you'll notice its spiral chloroplasts, which give it a distinctive appearance. This observation helps us understand how these algae carry out photosynthesis and their role in aquatic ecosystems. By examining Spirogyra closely, we can see the structure and organization of its cells, which is fundamental to grasping how these organisms function. Remember, this is a great example of how microscopic observation can reveal intricate details of plant-like organisms, connecting to our broader theme of understanding plant diversity and adaptations. So, take your time to observe the filament carefully and notice the key features we've discussed. Keep the moisture in the cotton to ensure the potato slices stay hydrated. Observe the changes over the next few days carefully. Now, if you look at the potato with notches or buds, you'll notice the growth of new plantations starting to appear. However, other slices of potato that don't have notches or buds do not show any growth. This highlights an important point about vegetative propagation. Certain parts of the potato, like those with notches or buds, are capable of developing into new plants. When we cut the potato into slices, Those with notches or buds are more likely to produce new growth, while slices without these features typically won't. This activity demonstrates how vegetative propagation works in potatoes, showing us that specific parts are vital for successful plant production. So, understanding these features can help us in farming and crop management, connecting to the broader theme of how plants can be propagated without seeds, just through their existing structures. Let's look at what happens when we keep one end of all these pieces in water. Observe carefully which parts of the plant show growth. As we can see, the stems of the money plant with leaves tend to grow, while the parts between two leaves don't show much change. This suggests that certain parts of the plant are more capable of growing than others. Now, let's try cutting the money plant into small portions. Notice that some pieces don't have any leaves, while others have at least one leaf. This activity helps us understand vegetative propagation, where new plants can grow from different parts of the original plant. It highlights the importance of leaves and stems in the growth process and how we can use this knowledge for plant propagation. Thank you for your attention. Observation. The seed contains two cotyledons, which classifies it as a dicotyledon. When it begins to grow, you'll notice the development of the future shoot, known as the plumule, and the root, called the radicle. Now, moving on to Activity 8.7, Observing Spirogyra. This activity helps us understand the structure and behavior of this fascinating green algae. Spirogyra is important because it plays a role in aquatic ecosystems and is a great example of plant-like organisms that thrive in water. By observing Spirogyra under the microscope, we can see the spiral chloroplasts, which give it a characteristic appearance, and understand how it conducts photosynthesis. This ties back to our overall theme of understanding plant growth and adaptation, whether it's in seeds or algae. So, I encourage you to observe carefully and think about how these simple organisms contribute to their environment. Thank you once again for your participation.